The gene-editing technology CRISPR may have been Nobel Prize-worthy, but Andrew Anzalone was convinced he could make it even better. Often likened to a pair of molecular scissors, it enables scientists to cut DNA and rewrite the code of life, such as turning off disease-causing genes. But the technology has some drawbacks. It breaks the DNA double-stranded helix, which can lead to unwanted changes in other sections of the code. As a postdoctoral researcher in the Broad Institute lab of David Liu, Anzalone set out to build the equivalent of a Crispr Swiss Army knife, a multifunctional gene-editing technology that can correct entire sequences of code all without breaking the double helix. The idea was to really try to broaden the scope of what we can do with gene editing. Anzalone says of the invention called prime editing. The human genome is made up of 6 billion combinations of the four letters known as bases A, T, G, and C. The bases make up segments called genes, which include the instructions for certain hereditary information. CRISPR-Cas9 allows researchers to hone in on a specific location within a gene and make a cut and add or delete genetic material. It is particularly good for inactivating or knocking out genes. But one of the downsides is unwanted insertions and deletions of random bits of code due to breaking the DNA double helix, which can cause unintended mutations in other parts of the genome. Other diseases, such as sickle cell disease, are caused by an error in a single base pair. That's where base editing, which can swap out individual bases, comes into play, though currently base editors can only perform 4 different swaps. This is where prime editing comes in. This technology can perform all of these functions, insertions, deletions and the 12 different mismatches of bases, and could potentially correct up to 89% of the known mutations that can cause genetic disease. It's being developed by Cambridge, Massachusetts-based biotech startup Prime Medicine. The company announced $315 million in combined Series A and B funding on Tuesday for the technology, which it describes using terms associated with word processing software. We like to use the analogy of search and replace, because the beauty of the CRISPR system is you can tell it where to go exactly in the genome. Our prime editing system also tells it exactly how to fix it, and that's really what makes it unique, says Anzalone, the co-scientific founder and head of the prime editing platform. The funding values the less than 2-year-old company at $1.2 billion, according to PitchBook. Prime declined to comment on the valuation. While $315 million may seem like a hefty sum for a company that really only started operations in July 2020 when it hired former Merck and Rhythm Pharmaceuticals executive Keith Gottesdiener, the majority of investors are repeat funders of Prime's co-scientific founder David Liu.